All right, now that we know what plasma membranes are built from and some of their functions, we're going to talk more about how things actually move across the plasma membrane when they need to do so. All right, so membrane transport, what is that um, actually referring to? All right, so as I've already mentioned, we talked about the barrier properties of the plasma membrane because of the those fatty acid tails that you have in there. We've also talked about how most solutes, dissolved substances in your intracellular and extracellular fluids are hydrophilic. And guess what? That means they cannot freely get across the plasma membrane. They don't like those fatty acid tails. They can't get past them because they hate them. They can't interact with them in any way. All right, so a lot of transportation, but as you guys know, if you're a cell and you need glucose for energy, and that glucose is outside the cell, well, guess what? Glucose cannot just wiggle its way through those fatty acid tails, but you still got to get it inside the cell. So many things are transported across the cell membrane through membrane proteins, like you see here, and some of those are what we call carriers, and some are channels. An example of a channel you see there, kind of like the name suggests, you got like a little tunnel through the protein that is sitting there in the membrane, and then a carrier is a protein you see over here that it's like a gate. It opens up on one side, captures something, and spits it out on the other side. And very often these channels and carriers are very picky about what they allow across. I mean, for example, you can have protein channels in a cell membrane that only let sodium across or only let potassium ions across. So they're very, very particular about what they allow to pass through the tunnel or what they'll carry across from one side to the other. All right, plasma membranes are selectively permeable, and that's just a fancy way of saying what we've already talked about. Permeability, the ability to permeate something, simply refers to whether or not something can pass across from one side to the other. And then plasma membranes are selective. They're choosy about what they allow across. Some things come across and some things do not. All right, so when we're thinking about substances moving across the cell membrane, we can divide those up into the processes that are used, can be divided up into two categories. Many of them are passive, and then others are active. The difference between these is passive processes do not require any energy input. Active processes occur when you have to force something to pass across the cell membrane. You've got to use energy, burn energy to make that happen. You've got to pump substances across. And we'll see some examples of each of these. Passive processes work because of the natural tendency for substances to move down what's called their concentration gradient. So a gradient means, all right, so if you had a whole bunch of a substance out here and just a little bit in here, and then you just kind of gradually go from more concentrated to less like that, that is a concentration gradient where you're going from more concentrated to less concentrated in one location or the other. And that's how substances tend to move. They want to move from where they're more concentrated to less concentrated. Okay, and the ability to do that, that type of function, is called diffusion. And we're going to talk a little bit more about diffusion shortly, but what determines whether or not some substance is going to get across the plasma membrane? What makes a membrane permeable or not to a particular substance? One of the key things is whether or not it is a lipid type substance, or It's capable of dissolving in lipids, interacting with those fatty acids in the cell membrane. That's lipid solubility, which is really another term for hydrophobic. Hydrophobic substances are more likely to be able to wiggle their way through the fatty acid tails of the membrane and get across. All right, now what if you're not hydrophobic? Well, then if you're going to get across, maybe the cell has channels or carriers that will let you come across, will let you pass across. Those function kind of like gatekeepers for allowing substances to move across or not. And so if a cell has, let's say, a carrier for glucose sugars, carrier proteins for those in its cell membrane, then it's going to be able to transport those across the membrane. If you don't have those proteins sitting in the membrane of a cell, that cell's not going to be able to transport glucose across. And so those things will determine whether or not cells can move things across as well. All right, there are 4 types of passive processes. So these processes do not require energy input from the cell in order to happen. And they're all based on diffusion. And again, diffusion is the tendency for substances to move from where they're more concentrated to less concentrated. And simple diffusion means that a substance can get across the cell membrane without any assistance. It can just worm its way across the phospholipid bilayer from outside to inside, or vice versa. That's simple diffusion. Now, in other cases, like where we were looking at those channel and carrier proteins, if a substance can't freely move across the cell membrane, then another possibility is that the cell has carriers or channels to help move those substances across. And that's called facilitated diffusion, because those carriers or channels are facilitated. They're helping diffusion take place. And finally, there's a 4th case, a special case called osmosis, and we'll have a separate video clip on that. That is the diffusion of water across a cell membrane. All right, so a little bit more about diffusion. Why does this happen? All right, so we already said diffusion is the tendency of molecules, ions, to move from where they're more concentrated towards where they're less concentrated. And so why does that happen? That's a very important concept in biology and other areas of science as well. Let's take a look at an example of diffusion. If you drop a lump of sugar into a little laboratory beaker there full of water, sugar is sucrose. That's a disaccharide. It's hydrophilic, loves water. So when you drop it in, first you've got all these sugar molecules very concentrated there in that lump, and you don't have any sucrose molecules out in the other parts of the water in there. You got lots of water molecules out there, but no sugar molecules. So your sugar is highly concentrated here, low concentration or not present at all farther away from that lump. So if you just let that sugar lump sit there over time, gradually those sugar molecules are going to evenly spread out within the beaker of water. That's diffusion. We've all seen that, seen things like that happen before. Why does that happen? The reason that happens is because all molecules have random motion. Even the water molecules, they're wiggling and moving around. Sugar molecules in a fluid are going to have random motion. So we can't see that. It's kind of hard for us to visualize. But all molecules in the air and fluid, wherever, are always in motion. So because of that, when you drop something like a sugar cube into a beaker of water, those sugar molecules are wiggling around, water molecules are wiggling around, and over time, the water molecules and the sugar molecules are going to even out inside that beaker. So that's why diffusion takes place. Now, how it looks to us is like things are moving from where they're more concentrated to lower concentration. And it's not really a conscious thing. The sugar molecules aren't saying, well, hey, I'm going to go over there where it's not quite as crowded. It's just that things tend to randomly even out due to the random motion that they're undergoing. Another way to maybe picture it is if you had a box and you put a big big handful of pebbles into a box in one corner, and then you just started shaking that box evenly back and forth, up and down, sideways, and so forth, to kind of mimic that random motion that molecules have, what's going to happen to the rocks when you open up the box? The rocks are just going to be kind of evenly and randomly distributed around the inside of the box. And so that's what happens with molecules as well. And that might appear to you like the rocks moved from where they were more crowded to where they were less crowded, but really it occurred due to that random motion. All right, so how does diffusion occur across cell membranes? All right, so if you have lipid soluble solutes, and again that just means they're hydrophobic, so things like lipids, we talked about in chapter 2 how those are hydrophobic. Those are substances that they love these fatty acid tails in here. They don't have any problem getting in there and intermingling with them, and so they can freely diffuse from one side to the other. That's called simple diffusion. Most solutes, though, in our body fluids, inside cells or outside cells, are lipid. Oops, screwed up with my pen there. Lipid insoluble. They can't dissolve in lipids. They do love the water-based fluids, though, and so they are hydrophilic. So how are they going to get across? So smaller ones, things like sodium ions, potassium ions, sugars, amino acids, small hydrophilic solutes like that, they can get across cell membranes through these carrier and channel proteins that we were talking about. So if a carrier is used, and again the difference is a carrier is like a gate. It'll open up on one side and capture something and spit it out on the other side. That's called carrier-mediated facilitated diffusion. And then if a channel is allowing some sort of small hydrophilic solute to move from one side to the other, that is called channel-mediated facilitated diffusion. And again, I know a lot of this probably seems pretty abstract and difficult to picture at this point during the course, but it will come up, especially when we get to the muscular and the nervous systems later this semester. All right, and finally, that last passive process is called osmosis. And now there are different things or the movement of molecules other than water can be referred to as osmosis. But for biology purposes, when you're thinking about osmosis, you're almost always talking about water molecules moving across a cell membrane. And so that's how we're going to use the term osmosis. So like they're showing you here in this diagram, water molecules can diffuse across cell membranes in either direction. Now that might seem totally contradictory to what we've been talking about. Hey, you've got all those fatty acid tails there in the cell membrane, and that's hydrophobic. It's oily. Oil and water don't mix well. But so like we said earlier, the plasma membrane is not a rigid structure. It's not like a concrete wall. So it's thought these water molecules, which are just H2O, oxygen with 2 hydrogens attached, they're not very big. And so due to random motion, they are not all that efficient at doing it, but apparently they can wiggle their way past those fatty acid tails and move through the phospholipid bilayer. Now also, some cell types will produce what are called aquaporins. Those are water channel proteins, and they'll have those in their cell membranes. And those, as the name suggests, aquaporin, water pores, cells that need to rapidly allow water to move in or out of the cell will make those proteins and stick them in their cell membranes to allow water to flow back and forth more quickly when needed. You'll hear about those when you study the kidneys and urine formation in A&P2. All right, so those are the different types of passive processes by which substances can move across the cell membrane. I'm going to split this video lecture clip into two parts, and on the second one we'll talk about the active processes that require energy for moving substances across a cell membrane.